Today I'm going to tell you about something we started, believe it or not, way back in 2019. Took us a long time to get this one done. And that is to look at the Formosa plastic discharge monitoring data. And I've got to thank Bill Harvey here, who helped me convince Formosa to give me the data. So if you look at it. And I'll start out with my thank yous. As you can see by all the little logos, it took money from a lot of different organizations to be able to pay for all the people who had to work on this. Let me go backwards. So this is me, and a lot of the work I'm going to show you today was actually a master's thesis for Elizabeth, who just finished a few weeks ago. And Audrey works full time with me, as does Rick Coffey here. And then Lisa Vitale and Dave Roseanne worked at Friesen Nichols. They've actually been involved in collecting the data over all these years. So anyway, Elizabeth, who really worked on this full time, like I said, since 2019, 3 years. Her entire time was funded by NOAA through an education grant we have at the university. And then we got some money from the water board back in 2018. That we were doing the Lavaca Bay assessment work, and that's when we first started trying to work on this data. And we barely scratched the surface of it back then. And then more recently, we got additional money from the Matagorda Bay Mitigation Trust to help us finish off the project. And so I think the first time I came up, I probably told you about something we had noticed way back in 2011. And that is that, so I study things that live on the bottom of the ocean. And the things that live on the bottom we call the benthos. And the reason why it's really good to study things on the bottom is because it's got a high diversity. And the funny thing about diversity is that when there's a disturbance or some kind of environmental change, the tolerant species will stay the same or increase, and the sensitive species will decline or disappear completely. And so the relative change in the different kinds of animals that live in the mud, it's literally like taking a pulse of the ecosystem. It's literally like measuring the bay health. And so that's our main assessment technique. And so what we noticed way back then, we had started sampling in '88. So this was 20 years worth of data. And we noticed that abundance, biomass, and the diversity were all declining. And these are log rates. Notice that it goes from 1,000 to 100,000. So it had averaged 20, 30,000 back in the 80s. And then it started averaging just a few thousand. And the same thing with the species. We went from getting an average of about 10 animals in the sample to just 1 or 2. And so that was an indicator that something's really, really wrong. And ever since then, when I say that, I mean ever since about 2011, we've been trying to figure out stations that they've been sampling for a long time. So, we knew that the data set was there. And of course, there's an industrial discharge that goes into the bay, and they have to monitor the discharge to make sure there's no adverse effects due to that effluent. And again, the interesting thing is this long-term decline I'm talking about, we noticed 5 years before they even started the discharge. So it's not like I can tell you they started the discharge and then it changed. That's not true. It was changing before that. So it's a pretty remarkable data set. And they've got about 16 stations. They go all the time. When we collected this data set, we got it through from 93 when they started to 2020. It actually is 112 trips. I'm not sure why I wrote 11. And they were measuring everything in the world. They were measuring biological the water column, hydrography, sediment stuff, and pore water. Pore water is very important. Pore water is the water that's in the sediment. The sediment acts like a sponge. And if you squeeze the sediment, you can get water out of it. And the thing is it's just like, the water in the sponge in your sink. You squeeze that water out, it's pretty stinky. So the bottom holds a lot of junk. And so that water in the sediment is really, really different from the overlying water. It's typically got a whole bunch of nasty compounds in it, and it typically can be toxic, maybe some sulfide, maybe some other toxic materials. So that's why looking at those four, we call those matrices. So, the water, the tissue, flesh fish, the pore water, and then the sediments themselves. So every time we measure a chemical, we measure the chemical in all four of those parts. And so the idea was, let's see if we can find out if we can distinguish between anthropogenic, meaning man-made change, and natural change. And in particular, if we can understand a synergy where two different things, each one by themselves wouldn't cause a problem, but when you add them together, they do. And that was the goal of the project. And again, we started way back in 2019. And this should look very familiar to you. Here's Vaca Bay and the point comfort facility here. And the discharge, it's a little hard to see it. It's right there where this big red star is. That's about where the discharge point is. And as I said, ironically, and this is their whole sampling design, but ironically, if you look at the inset, this inset is a blow up in that area. You can see I've got this station A that Rick started sampling that way back in 1984. So we had data at that station A going all the way back to 1984. And ironically, they put the discharge pipe right next to it. And that's how come we would run into them when we're out there sampling, because they're out there sampling in the same spot. And then just below the bridge, we had another one we used to call B. So we have these stations that are a little bit further away. And if you look at the way you can see it a little easier on this side, the way they set up the samples, it's like in rings. So you got the A ring, which is really close to the diffuser, then the B ring, and then the C ring, and then the D ring, and then you go farther away, and you have the R ring, R for reference. So the bottom line is, it's what we call a classic bullseye design. It's kind of like a dartboard. And the whole idea is that if there's any pollution coming from that source, then it should decline as you go away from the source. And at some point, you should get in the natural background and not see any effects from whatever that discharge would be. So the main thing we're looking for is, are there changes with distance from the discharge? That's the whole idea. And, oops, I keep pushing it the wrong way. And these guys collected everything in the world. And I'll be honest with you, I've never seen such a comprehensive study. It's amazing. They measured every kind of organic contaminant, metals, and pore, saline chemistry, water chemistry. We had necton, which is like fish, and epifauna, which is like the shrimp, crabs, things that swim along the top and bottom. Ichneoplankton, or the baby fish, the fish babies swimming around in the water. And then the phytoplankton, are the small plants, that all the zooplankton that live in the water column are feeding on. And then we're looking at benthic data as well. So the bottom line is they were measuring every single trophic level, which meant we could do something else. We could actually do a systems level analysis when we're done. In other words, try and understand how are the parts connected to one another. So it was pretty exciting to get a hold of a data set like this. But it was huge. That first dump, Lisa Vitale sent me 1,223 files. And as you might imagine, it took a little time to wrestle those things all together and to put them all together in the first place. And they were in like 4 different formats, so that was a whole nother thing. And since the early ones are done in the early 90s, luckily I'm a computer geek, and I actually had software on my computer where I could read old stuff that you wouldn't be able to read anymore if you weren't a computer geek. And we got that, believe it or not, in August 2019. That's how long ago we started working with this. And as I said, we just finished our first draft of a complete analysis only in April and May. It took a total, we didn't finish everything, wrestling that data set in shape. I didn't say it's done, it's baked, take it out of the oven. I didn't say that until February 1st, 2022. It took us 3 years to wrestle this baby into shape. And one of the things we discovered, and it took us a year to discover this, was that 5 of the trips weren't even in there. We had to type all that in ourselves. So after we typed it all in, and I convinced myself we actually have all the data, all the numbers that are in the written reports or in the files, then we put it all together, like Humpty Dumpty getting put together again by all the king's men and all the king's horses. And then we started discovering weird things. We do what we call quality checks. So for example, I know that the bay is never going to go between maybe 55 degrees bay water, 55 degrees Fahrenheit, maybe 93. So we run a check and we say, are there any bay temperatures that are 120 or 32? Because if there are, I don't believe it. And we would do that for every single variable. We would set up a range and say, is there something in there that just doesn't look right? And then we'd go and look at that number, compare that number to the reports, and every single time we found that was an error, usually like a decimal place got put over. So something like 32, 3.2 might have read as 32 instead, And after we started finding hundreds and hundreds of those things, that's when I realized, oh heck, we've got to look at this. We've really got to do a complete quality control check on the whole thing. And we literally, it took us about 4 months and had about 4 or 5 students do it. We started this last fall in 2021. We finished it right about Christmas time, maybe Thanksgiving time last year. And we checked every single darn number in that data set. We checked it. We did it. And then we rebuilt the data set. We run all the computer programs to put Humpty Dumpty back together again. And when we got finally done, the data set had 672,000 rows and 24 columns. You multiply those together, that's over 16 million cells. So we had a data set with 16 million bits of information in it. And that's what I mean, and that's why it took us nearly 3 years, 2.5 years, to just put it all together in one place. And the good news is, that's all available online right now. I put the whole thing in, we have a data repository at Hart called GridC. And if you know what a DOI is, a digital object identifier, you can type that into Google and the data set will pop right up. And you can download it and play with it yourself. But in it, there were 750 chemical parameters. Again, that's in the 4 matrices. And 1,326 species. So there was information about a lot of stuff. So the question then is, what do you do with all this stuff? The first thing we did, as I pointed out, I was most interested in the contaminant data because I want to know, are contaminants playing a role in the long-term decline of the bay? And here was a huge disappointment. So these are the contaminants of what we call a physical chemical, and these are the percent of the samples that were non-detects. And if you look at it, when it comes to all of the toxic chemicals, PAHs, which are like oil-derived products, PCB, polychlorinated biphenyls, that's another industrial man-made chemical, pesticides, 2 other kinds of organic contaminants. The bottom line is there was actually no data in the data set. Nearly 100% of them, 99.9 times, they would get a non-detect, which meant out of those 400,000 measurements, there was no data. So that was a bit of a disappointment. And the reason was they used techniques that have a very high detection limit. And I'm going to tell you more details about that in a minute. But for most of the important things, we were able to find data. By conventional, we mean like salinity, temperature, the normal stuff you would measure. The only thing that was low were the nutrients in the water column. Most of that stuff was non-detect as well. Luckily, we have our own nutrient data that we've been collecting for the last 30 years. So that was the first thing we learned. And in some cases, it's a problem. So the detection limit that their lab was using, particularly in the sediments, and the sediment is where most of the pollution is. And that's because of gravity. And in fact, I always like to say the bottom is like the memory of the ecosystem. Because everything's going to die, and gravity and everything's going to follow the bottom, and there's a record of everything that's ever happened through time in the bottom sediments. Again, that's why the bottom sediments are good indicators. And the problem is, for at least these 5 PAHs, this number is called the effects range low. This is a number established by NOAA, the National Oceanic and Atmospheric Administration, where they know above that number it causes biological effects. And so there are several important PAHs that can cause a biological problem, but they were below the detection limit. And these are the EPA methods that were required by TCEQ and EPA. They were the ones who set this standard. But the good news was all the metals had a detection limit above the effect levels. So if there's a problem with a heavy metal in the environment, we would be able to see that. And we know that there are PAHs that are toxic in the bay, and we know there are PAHs that cause a problem, because a guy named Scott Carr, someone I used to work with quite a bit, by the way, he had done a sediment quality assessment, way back in '99, I think this was, and he found plenty of these in the sediment, whereas in our data set, there's none. And the difference is the EPA method 8270 is used, whereas they use the NOAA ambient technique measurement. And the bottom line is the measurement limit in the current study was 150, but when you use this ambient measurement, it's only 0.5. So there are methods that are 300 times more sensitive to find pollutants in the environment. And the good news is we just finished a sampling event 3 weeks ago, where we sampled all of Lavaca Bay and all of Matagorda Bay, and we're going to do the same thing Carr did, but we're doing it at this level. So if there's anything there, we're going to find it this time, hopefully. But the bottom line is, again, that original study by Carr, he was primarily looking at the Mercury Superfund site, so nearly all his samples were below the bridge on the other side of Highway 35. He had one station by the diffuser, and it, by the way, was the most toxic station in his study. But anyway, the next thing we did was a time series analysis. We wanted to know, are things changing over time? And again, I showed you my data set, which was benthic, and the only ones that are changing significantly are dark, and it's less than 0.05. That's what we call our significance level. So this is a statistical test where we want to know, is there a trend in the data, and should we believe it, or are the numbers just a bunch of scatter? Just a bunch of scatter, there's nothing you can really see. And the bottom line is, they really weren't, we actually saw increases in zooplankton, but the only thing that went down, let me see, the only thing that went down, see here's negative, these are going down, was in the trawls. So the nekton, which is a fish, the kind of fish they catch in trawls, is the only other thing that was going down over time in their study. Everything else pretty much stayed the same over time. And then the next thing we want to do is, well, what's happening with, like I told you, the metals, those are good detection limits, so we can check that and have confidence. And look at, R is the correlation. Correlation goes from +1 to -1. If it's 1, it means it's a perfect correlation. If it's -1, it's an inverse. It's going down, not up over time. And if it's 0, it means there's no change. So the closer to 0, it means nothing. You can see all the metals went down over time, which to me was a big shock. So let's look at one of the important ones, mercury. Oh, here's something else that was real interesting. The difference between sand and silt. So sand was going up over the time, and the amount of silt or mud in the samples were going down. So that meant that sediment structure was changing. I'm going to tell you in a few minutes why that's so important. And then there's a bunch of other things, nothing too interesting. I want to tell you about mercury. So again, this is mercury over time, and remember the effects range is 0.15, which is right about here, okay? And the bottom line is early on, and you remember there was a lot of mercury contamination in Lavaca Bay back in the 70s and 80s. And early on, you can see there was some pretty big problem. We were well above the toxic effect range. But really, since about 97 or so, 96, 97, we've mostly been below that level. And also, you can see it's still coming down a little bit over time. And this is a trend we see on the other side of the causeway as well, over on the old Superfund side. So the bottom line is, it looks like this problem's cleaning up. So far, the only real contaminant information we have, because we don't know about the pesticides, the PAHs, the PCBs. We do know about some of the heavy metals, which can be toxic. So far, all of this is positive. This has actually been going down over time. So clearly, it's not coming from the discharge. It's the legacy that was there that's getting buried with new sediment. And that's what happens to heavy metals. They literally sink through time, and they're not available to the critters on the surface. So that's a good sign. But we saw some weird things when we started looking at the biological data. So again, this is a log scale. So 3 means 1,000, and six zeros means million. And what we noticed was somewhere around 2009, 2008 actually, there was like a 3-fold jump. And when we asked the people at Freeze Nichols what's going on here, they pointed out that they changed their methods on how they were counting. So it's hard to look at the phytoplankton data and understand what's going on with primary production, because they kind of changed their methods in the middle of the study. Ironically, the bottom graph is always going to be the species diversity. It actually stayed pretty common, pretty similar. You can see it's got a similar number over here, around 40, 50 things in the sample throughout the whole study period. So that also is a relatively good sign. Bottom line is primary producers don't look like they're having a problem. And with zooplankton which are the critters in the water that eat the phytoplankton. Of course, little fish eat zooplankton, big fish eat little fish, right? So this is all part of the food web now. It also had a weird pattern, where they were finding 2 is 100, a couple of hundred up until about 96. Then all of a sudden, between 97 and 2009, they were finding about 10,000. And then all of a sudden, after 2009, they're finding 1 million, almost 10 million in every sample. I don't know what that's about either. They couldn't explain it. I just felt like we couldn't really use this data also in analysis because we know something's screwy, but we don't know why. And maybe due to changes in subcontractors. This has been a complicated project over time. They've had many different subcontractors come and go. The same people have been in charge. The company name changed 4 times, but it's been the same people from the beginning. But they have used different subcontractors in some cases. So we didn't feel like we could use all of that data as well. But again, it's interesting that if we look at just community structure and diversity, that really didn't change much, maybe a little different with this bunch, but after that date, it looks pretty good. So we're not seeing a big change over time there, and that looks reliable. And we could look at all the other things. Everything goes up and down over time. The important thing is none of these have a downward trend. That's what I want you to see here. There are no downward trends in anything else. And except, and I pointed this out earlier, except for the trawls. The trawls show a clear downward trend over time. Look at that. Here's the total number. And again, this is logs. This is 1,000. This is 100 in a sample. And it's definitely changing over time. Back in the early 90s, it was definitely always around 1,000. And now it's only running in the hundreds. And hell, this is 10. You know, here's one sample with virtually 0. And the bottom line, and the number of species have been declining as well. So that's the only thing where we're seeing the big declines is in the nekton, in the fish. And I know you guys tell me, I've been having problems with fishing over the last 10 years, because that's what this is indicating to me. And so that's what they found in the trawls, and so that's a problem. So the next thing we looked at is, well, is it natural variety?
Yeah, when you mentioned the trawl, would that also include shrimp, or is it just?
Yes, the trawls would have shrimp.
Yeah.
Shrimp, crabs, flounder, bottom fish.
Shrimpers have remarked about that.
Yeah, shrimp, crab, bottom fish, like a flounder, things like the redfish, black drum, and trout, they would find them in the gill net. The gill net samples. So this is not the big net time. So the next thing we did was, well, let's look at the hydrology, river flow, and climate. And there are 2 interesting stories here. So the top graphs here are the USGS gauge on the Lavaca River. And as you can see, there's a pretty strong cycle of wet years and dry years, right? You can see a really strong, deep drought between 2010 and 2014 and 15 or so. You can see some big floods. You see Hurricane Harvey over here. No, that's not Harvey. Actually, you didn't get much of a Harvey signal up here, did you? And what we did is we took the total amount of inflow for 30 days for every sample, and we ranked it, and we came up with this number, and that allows us to categorize periods as dry, wet, or average in the middle. So we're able to look at wet years, dry years, and average years. And then when we looked at just temperature, cumulative, so this is cumulative inflow by month, that was very interesting, because we definitely get more rain and more inflow in that April, May, June, and again a little spike in October, which I'm guessing is hurricanes, right? But notice the error bar Look, notice how big it is in October. That's because some years there's a lot more variability in the fall. So the fall, you can get a lot of dry years, you can get a lot of wet years. Whereas in the winter, January, February, you're going to get mostly just dry times, okay? And on the right is temperature in the bay. Darn it. Look how small the error bars are with temperature. So the bottom line is the temperature has been pretty consistent every year if you average it over a month for time. But the other bottom line is there's not only a strong seasonal signal with things like temperature, but there's also a strong seasonal signal with inflow, and there's year-to-year variability. So this is one of our big hits. And we have a statistical technique we call principal components analysis. So we put all the variables that we measure in the water together. And if they're correlated, they show up in the same place on a graph. So what this graph is showing you is that when salinity is high, turbidity and total organic carbon is low, and when dissolved oxygen is high, temperature is low. So basically we have this new variable we've created that's an indicator. So we have a variable that explains seasonal variance, which would be the x-axis, and we have a variable that explains freshwater inflow variability, and that's the y-axis. And so now we can use those two numbers, and we could figure out, are the changes we see due to the little bit of metal contamination around, or is it due to these natural climatic events? And so the next chart, we take every sample now, and we classify every sample. And on the right are the different stations. And the bottom line is you can see every color is all over the place. That means the discharge is having no effect on the overlying water column. That's what that means. But when we look at when we plot as symbols wet, average, dry, you can see on this inflow axis, you got almost all D's, and when we go negative, it's all W's. So now we can classify every single sample as being a wet year or a dry year. And then the other thing we were able to do is classify seasons. So winter is 1 and fall is 4. And you see I have all your 1s on the right and all your 3s and 4s on the left. And so again, we can look at a biological response now, and we know exactly whether it's being caused by year-to-year differences, wet year, dry years, seasonal differences, winter, summer, and we can correlate it to chemicals too, and see if the bottom sediments are playing a role. And we do the same thing with sediments, and the important thing here is this is the first real strong evidence that the discharge is having an effect on the bed. So these are the stations, and what you notice is, like, all these sort of red squares are here, all the circles are up there, the diamonds are over there, and the squares are kind of in between. So the bottom line is, we're starting to see some change in the sediment with distance from the discharge. And we also noticed that there was a strong change in sediment over the years. This is the early times, the middle years, and the blue is more recent years. So the sediment has been getting sandier around that discharge. So when we look at the biology, what we do is we ask, again, I want to know, is there a difference with distance from the discharge? And remember, A is close, B is a little bit far away, C is further away, and R is furthest away, okay? And if you look at this for the benthos, you'll see that, wow, all the R's are pretty different from in close. So you got different communities in the discharge area, in the sediment, then away. And also, you can see all the A's right here are kind of grouping together. And so we're also seeing a difference with distance, because you have your A's, your B's and C's are the same, and then the R's. So you see a real clear gradient that the community is changing as you move away from the discharge. And you gotta assume that R would represent the natural background, so this is a change in the natural background. You'll notice for the trawl and the gillnet, the same thing happens. The R's are out there by themselves. The gillnet samples, boy, they all clustered right on top of each other. All those, well, there was an A, well, actually for gillnet, they didn't have A's, they had only B's and C's. They're all just exactly the same. And I'm trying to remember the number now, something like, it's real close. This area is not even as long as this room, okay? That's why the samples don't look very different. And if we look at the trawl, we see a pretty good scatter again with the C's and the B's. So the bottom line is you go from a B to a C to an R. You can see a transition over space, right? It's kind of going, you got your B group, your C group, and then your R's are really different completely. So the bottom line is we found some evidence that these biological changes are occurring with distance from the discharge site. And when we look at the water column, we saw the same thing, which shocked me really, that the R's were different for the phytoplankton, zooplankton, and even for the ichthyoplankton. I didn't expect to see that at all, but we still saw differences there. In the water, they only measured mostly the B ring and the R ring, so we only have 2 rings. The problem is, when you only have 2 stations, you have this problem we call pseudoreplication. In other words, if you sample any 2 places, they're likely to be different just because they're 2 different places. And you don't necessarily know if they're different because of why you think they're different. In this case, it's near the discharge and away from the discharge. So technically, because there's only 2 stations, we can't actually say, and that's because of discharge like it could with the other ones. And the other ones, we can show a clear change with distance away. So we know that it's related to the discharge. So, like I told you, the other thing we can do is a system analysis. Because we think that, the sun and the nutrients are growing the phytoplankton, the zooplankton are eating the phytoplankton, little fish are eating the zooplankton, big fish are eating the zooplankton, the little fish, and maybe you've got some other fish eating on the bottom. Particularly, you expect things that live in trawl, shrimp, crab, flounder, to be eating the benthos. So the question now is, can we link those things together? And so we use a statistical technique for that. And the bottom line is that, what we found was most important. You can see the little double stars. That means this is important. So inflow is driving salinity, no duh, right? What was surprising is it wasn't really affecting, we only had 2 nutrients that were measured, and it wasn't really having a strong effect on the nutrients, but the amount of DON and phosphate in the water, you can see the 0.92 was also affecting the phytoplankton. So the bottom line is, the inflow is bringing in nutrients. The nutrients are making the phytoplankton grow. And then we've got the zooplankton come in, but they didn't seem to care. Look at that number, 0.01. So there's no evidence that the zooplankton are eating the phytoplankton in this system. Instead, the zooplankton seemed to only care about the temperature. The phytoplankton did. The zooplankton didn't seem to be responding to either salinity, temperature, or phytoplankton, because there's no stars near these numbers in the arrows. The other thing that was important is how much temperature affects dissolved oxygen. And this is something we know. In the summer, it's hotter and it's saltier. And the solubility of oxygen in the water goes down. So in the summer, you typically get low oxygen, and that's bad for organisms, because the organisms breathe as well. And so what we're finding then is that the seasonal dynamics are related not only to temperature, but also the relationship to oxygen, because in the summer, the organisms run out of oxygen to breathe, literally. And so the next thing we did was we built a sediment model, and it's got the same kind of thing. Again, inflow drives salinity, but look at how important salinity was. It's got a double star for the trawls and for the benthos. So that salinity was really driving it. But look at this. Not only do we have double stars, but we got some of the highest numbers on the whole graph. That change in the sediment, the amount of sand and clay that's changing over time and changing with distance from the discharge, it's getting more sandier over time. That's really what's driving the benthic change. So it is true that the discharge is affecting the bottom living critters, but it's not through what I thought it would be, which would be some sort of toxic or pollution issue. It's because the sediment around that area is changing. And again, temperature is really important on all these things. So, as I said, they didn't find any decline. We've recently finished up a whole bunch of our own samples. Remember I talked about that Station A, just a few yards literally from where they were sampling. And we continue to see a long-term decline in benthos even today. Remember, the study that started all of this stopped in, I think, was it 2008, Rick? Yeah. It stopped after 20 years, so it stopped about here, 2000. That was my original study from year to year. And now we can see it really hasn't come back. We get a few peaks, but for the most part, we're still seeing a steady decline, in my sampling at least, in Lavaca Bay. So what's the punchline though? Oh, the other thing is when I look at my data set, we got the sample A, which is close to the discharge, B on the other side of the bridge, and FD would be right at the diffuser. And we see a definite trend as well. And this wasn't true when I first told you about this several years ago, or a year ago, but we've got twice as much data now. We only started sampling there and working there in about 2008. And you know, the abundance is definitely going down from A to B to FD, and the biomass is going down, but the diversity is pretty much the same, right around 4 or 5 animals in a sample. So the bottom line is we are seeing in our data those two things. The bay still seems to be going the pot. And we see a tiny change related to the discharge, but it's hard to say it's being driven by a discharge because it looks like the real driver is going to be that salinity temperature issue, kind of the natural cycle of the season, so to speak. So, what did we learn? The freshwater inflow events, the year-to-year variability, and that seasonality seems to best explain all the change in this entire data set. Just seasonality. The discharge is definitely having a small effect on ecological condition of the bay. It's mainly affecting the bottom, the things that live in the sediment, the things that live in the sediment. It's a shame that 99.9% of all of the contaminants were non-detects because we still don't know anything about contaminants. So is it affecting? And the answer is, we're doing another study right now to try and figure that out. We're actually measuring the contaminants, and we'll measure diversity and toxicity of the sediments. And the one thing we have found though, again, this is at that FD station we've been sampling. A couple of years ago, Rick came to me and said, all of a sudden these little plastic beads have showed up in all of the samples at that discharge site. And these things are ranging between about a 0.5 millimeter and about 1 millimeter
They're right at about a half mil.
Yeah, right about a half a
And
million.
a few of them will go through a half mil and get caught on 125, but our cutoff point's a half mil as
Yeah.
far as our sediment, so that is mainly what has been picked up. And that's just a conglomeration of several samples. Right now, we're actually enumerating them, or
Yeah,
quantifying
we
them.
count everyone now.
We're counting everyone, once they started showing up and say, well, it's obvious that it's there and it's not at other stations.
Yeah, we never find it.
But we'll talk about that later, and it'll come out.
And we actually sent these off to a lab to get them characterized, and what did the guy say they were, Rick?
It's part of a catalyst system,
Yeah,
as far as plastic production. That's what those are used for, it's a catalyst.
and so we have a chemical name, which I can't remember, I forgot to write it down. But the bottom line is there is a marker, and they vary over time. It's not the same all the time, by the way.
And it may be that, like Paul's saying, it could be that we get real high concentrations sometime, and just recently, the numbers have dropped off.
I noticed in the last one you gave me, there was virtually nothing there.
They've dropped off, and they were in the 50s to 100s per sample.
Yeah,
And now they're down to 6, 7, 8, 9.
Right.
And it could be, you look at an inflow effects or something, moving the sediment around. And for the longest time, we didn't get any at Station A, but we've actually picked some up. I'll say that's what,
yeah,
a half a mile or so away
Yeah, half a mile away.
from this. And we're picking up one or two or so at Station A, so it's not like they're staying right there. They get rolled around, dispersed. But you say, you look at the nurdles or the flakes or the stuff that's on the shoreline, that stuff is buoyant, it gets up on the surface, it's blown by the winds and the currents, whereas this stuff sinks to the bottom.
it was all sinking.
This actually is in our bottom samples, it's not something that floats around. But I think it can get washed around if the
Yeah, it
currents.
sloshes back and forth with tides or with a big flood pushing downstream.
Another thing that we need to really, I feel, look at, I know when you do analysis, we need to look at some of the 0, 3, 3 to 10. That 3 to 10 sometime is black here, or
Yeah, so-
H2S.
The reason why we find fewer animals near the discharge is because they don't penetrate the mud as deep. That's what Rick's talking about.
Typically, you have higher numbers, smaller biomass on the surface, 0 to 3, and we sample below that, you get larger biomass and bigger animals, but fewer numbers. They're longer lived animals, so they don't really penetrate or come up. But on this site, I have a feeling that if we do the analysis on that, we're gonna see some differences.
Yeah,
And we need to compare that with 15,
yeah.
because there are some differences in depth.
So, no one's asked me for my opinion or my recommendations. But I'm gonna just tell you what I think. Anyway, it seems to me Formosa is wasting a lot of money. And, maybe measure a few things with better techniques. And in particular, rather than measure everything in the world, things that, there's no reason to think pesticides are coming out of that pipe. What are the targets? Identify indicators. The kind of chemicals that are in this plastic bead, for example, this catalyst bead, for example. Why don't we just measure fewer things, measure them with more sensitive methods so that we can really start understanding if there's a contamination issue. And that's basically, what we're saying here. There's almost no point in all the money they're spending on that contaminant analysis. When 99.9% come back non-detect, that's never going to change. You're going to have to use much different techniques that are designed for the regular water, not a discharge area per se. And, the strongest changes we're seeing is in the sediment, so focus on sediment's important. But I really think, you can't have just two stations, because when you do, all we know is that the two stations are different, but we don't know why they're different, particularly if we don't have an indicator measure. So if two places are different, and the contamination levels are very different, you got something. But because we have no contamination data, we don't have anything. I think they need some stations between R and B rings closest, so they got a lot of stuff bunched in real tight, and then something really far away. And to really understand if there's this change with respect to distance, probably could use one in between. And the other thing I didn't mention is, they never replicate anything. They measure just one sample at every station for everything. And that's a problem because you can't compute statistics. So, again, less is more. Measure fewer things, but measure them with better lower detection limits, and get more samples, replicates. Because if you don't have multiple replicates, you measure the same thing 3-4 times in one place, then we know are those two places different. Because what happens is you get a range. So if the range is this big in one place and it's that big in another place, you know they're different. But when you have just one point in each place, there's no way to know if they're different or not. So they both can be within the normal range of error, or they might not, and you just don't know. So, and this is not Formosa's fault. This whole program was designed by TCEQ. And so it's really a TCEQ problem. And I don't know if there's ways to change the sampling plan or the design, or how long you're going to continue. I don't know anything about those details. But someday I'd like to talk to the guys over from Formosa about that. And I just think for the money they're spending, they can get a lot more bang for their buck. That's just my thoughts.
I know we didn't get involved in this actual sampling and discharge until I ran into Diane. She gave a talk in Corpus, and then I got her book and read her book, and then we got her down for a seminar, and that kind of rattled my cage. And I just added Formosa discharge to our sampling program, and I started sampling it. And I think that over time, we're gonna see some differences there.
Well, I just showed them to you.
Yeah, there will be some differences.
And when I was here, I think 2 years ago, I had maybe a third as much data, and it wasn't looking like they were different, but now that we have more, we're back to, when you have a lot of samples, it's called power. Your ability to detect change goes up. So that's why lots of samples are always important. There's this old saying in science, how in Hollywood they say you can't be too rich and you can't be too thin? In science we say you can never have too many samples. That's my joke. That's what's called a geek joke. Anyway, guys, thanks a lot for listening. If you have questions or comments.
Well, I don't know if you saw that water quality data, and you can clearly see over time an increase in salinity temperature.
Yes.
And about 1 degree centigrade or so, maybe 1.5 degrees centigrade. And I remember before I started looking at all that data, reading some scientific publications, and one of them talked about the energy flow into that system being principally allophonous.
Yes.
And so we looked at Texas Parks and Wildlife shrimp trawl data. Well, we're looking at, mostly we're looking at brown shrimp. That was the species in question. And what we were finding is that particularly after the mid-90s, when I started doing some of the license buybacks, the numbers started to go back up, but they were all smaller. So when you think about, and then looking at the distribution of shrimp catch and oyster catches around Vaca Bay, up in the Tres Palacios area, Powderhorn, even over Chocolate Bayou, they're higher. And it seemed to me that the one thing that those three locations had in common, that's not in Upper Vaca Bay, is those streams are not dammed. And so my question for you is it possible that some of the changes we're seeing is that when we put higher temperature, higher salinity, lower oxygen, and maybe infrequently or frequently lower amounts of energy entering the system. You know, all of those things are synergistic to producing smaller animals and less of them.
That's pretty much exactly what I think is going on. And we actually have done a few studies showing that in the other base systems as well. And we did notice the same thing. Over time, I think the last time when I finished the Lavaca Bay Assessment Report, which was in 2018, I think that was my first time I came and we pointed that out, that there has been a long-term increase in salinity. It didn't show up in this data set, but we know from other data sets it's real. And we also know there's been a long-term decrease in inflow. More water use, more water diversions. This is happening everywhere in the state of Texas. And it's a little bit of that. The water temperature in all Texas bays and even in the near shore of the Gulf, has been increasing since about 1970, 1980. And again, with increasing temperature, increasing salinity, that's driving dissolved oxygen down. And the fish and the little worms are just like you, they need oxygen to breathe. And they start out with a huge disadvantage. We breathe air, which is 20% oxygen. They breathe water, which is only 7%. So when oxygen goes down a little bit in the bay water, it can cause a real big problem real quick. And that's something we've seen for sure. And when I started, I said, at the end of the day, I think this is a synergistic problem. I think it's a lot of little things. Each one by themselves wouldn't be a big problem. And when you investigate them all individually, you don't really see it. And that's why we try and do that system diagram I was showing you at the end where we connect all the dots. And that's what the dots are saying, exactly what Bill just said, that there's a real strong salinity and temperature signal. The salinity is driving a lack of nutrients and when it rains, stuff in the watershed flows downhill. It picks up nutrients, it picks up twigs, it picks up organic matter, that washes in the bay, that fuels primary production. And again, that gets the food web going. And what we're talking about is something we call oligotrophication. In other words, when we reduce the flow of water into the bay, we're also reducing the nutrients and the organic matter in the bay that the critters need to live on. So the bays are definitely becoming more store for food. And they're becoming more stressful because oxygen is going down and temperature is going up. And then of course there's probably some contaminant issues. But right now it looks like those big two things are the big drivers, but not the contamination.
So water use and not enough inflow can look like a real trigger. It's like, so what about this new water use by the Lackawanna River Authority, 2,500 acres on Promosa's property to divert water to? I mean, is that, would that?
Well, it'll just add to the problem. So, a whole nother process that's been going on since 2007 has been that Senate Bill 3 environmental flow requirement thing. And as I recall, the standards in this base system were developed in, was it 2011 or 2014? 2013, something like that. And the standards are supposed to be revised after 10 years, but no one is doing that. In fact, probably because of COVID, the groups that are supposed to be looking at these inflow issues haven't even been meeting. The Colorado Lavaca group was really good. They were probably meeting more than any other one in the state. They were real strong, in my opinion. But again, because of COVID, things haven't been happening in the last 2 years. And considering that we're supposed to, according to the law, be reevaluating those environmental flow standards, we probably need to kind of kickstart that again and get that process going again. I think that's probably the most important thing statewide, by the way. This is true in every basin in the state. Some basins are better than others. And again, I would say the Colorado Lavaca Basin, which has been led by the guys like at the Navidad River Authority. They've been doing, in my opinion, a good job of trying to keep the process moving. In other places, the river authorities have been terrible. They've totally abdicated. For example, in my backyard, the Oasis River Authority hasn't held a meeting in maybe 7 or 8 years. You know? And when we try and kick them and say, hey, you know, we need to get on this, they just ignore us. So, you know, it varies quite a bit in every single river basin, but it's something we definitely need to kind of get back into and push again. And, you know, at the end of the day, Texas is always going to be a water limited state. And the more west we get, the drier it gets. And because the whole coast curves to the west, the further south on the coast you get, you start running into problems. And that starts right here, by the way. You go north of here and they get just enough rain that, like Galveston Bay, believe it or not, actually has even a little bit more inflow than it did historically, because they pump groundwater. And then, you know, use it for your shower and your toilet, and then it goes into the sewer system, and eventually that goes to the bay. And believe it or not, the Galveston Bay system actually probably has better flow than it did historically. But that's not true anywhere else in the state. And that's particular, and it really starts right about here. And of course that extends all the way down to Corpus Christi Bay and Baffin Bay. And yeah, I think this is one of the biggest things we're facing. And as I said, I think it's a lot of little things. It's not like I can say, if you do this one thing, we'll fix it. It's gonna take, you know, and how much of that little thing do you have to do? Those are things we're still working on. Well, I think the important thing is I think we're finally getting a handle on exactly what it is. And here's the good news. There's really no big indication that there's a big contamination problem, at least in this system. The problems here do seem to be related more to climate and water diversions. And I think that's pretty common in the other bays of the state. Yeah.
Are there any studies as far as like the shoreline health, you know, changes in the Spartina beds and, you know, if rising sea levels, you know,
Yeah.
are knocking that down or?
Yeah, a little bit. You know, one of the things that's really important was both the marshes and the oyster reefs. Those fish love those edges. That's the prime habitat in the whole bay. And so, you know, as you, and for the most part, those sorts of habitats, the marsh habitats are doing okay. Oyster habitats, of course, they get fish, so they're not as good a shape, and they're changing over time for sure. And there have been studies, you know, showing the habitat utilization of those things. There's concern about sea level rise and marshes drowning, literally, as water comes up. But I have to say that those sorts of studies, you've got to be careful because, you know, the rate of sea level rise is very slow. I mean, it's noticeable over 30 years, but year to year, you're talking, you know, maybe like a fingernail's worth. And the point is that the plants can keep up with that. So whether or not plant cover can keep up with sea level rise is going to depend on the way the shorelines are configured. If they're natural, they'll probably have no problem. But where we build walls or jetties or, you know, where we have a steeper bank, then it'll be a problem because they won't have any place to expand to. So that's the sort of thing we think is going to happen in the future.
Can I ask one question?
Sure.
Another opinion. You know, when you kind of look at the water flow through Walker Bay, Patagora Bay, it almost, in talking to people who've been here for much longer than I have, they talk about how when you get really high tides, they don't stay high as long as they used to.
Ah.
And that, so it made me think that again, is also residence time of water in the system have something to do with that in terms of, think about getting nutrient inflows. If it doesn't stay in that system for a long enough period of time, it can't be assimilated, or be assimilated at a slower rate if it has a lower residence time. And again, when you think about how deep the channel is right outside and that's eroded away very rapidly, and there's a tremendous amount of water going in and out of there. So could residence time perhaps have something to do with it?
Yeah, so yeah, this is actually one of the main concerns about deepening the ship channel. That was one of the big things that were brought up in all those meetings we've been to on the ship channel dredging, concept. And, I'm not too, I haven't seen that information about, the water taking, it goes out quicker?
Yeah, you see, it just doesn't have the residence time. If you think about it, going through Pascagoula historically, as opposed to coming straight out of, almost a large tube, relatively.
It
Yeah,
seems
but-
like this bay system has a quicker exchange than what, say, Corpus Bay does.
Yeah.
The residence time in this bay is a lot quicker. I don't have the data to say
It's
that.
about 10 times
But
quicker.
just from what I've seen working here,
That I know.
just like when we started that project in '84 to '86, the water board, had us go up there and work, the upper part of Oahu Bay, and people were complaining about the high salinities upriver. Well, they had a big release to take care of that. We had a storm or something come through, and all that was just flushed back out, and the salt went right back up again. It's not like the release did much of anything. It did for a short period, but naturally, this bay, from the pass straight up to the back of the bay, it's a quick exchange. Like I've seen sargassum before floating around our Station B up there, triple tails up there, to me, there's a big exchange in this bay compared to other bays.
It doesn't seem like there's as much tidal influence as you get further up in the bay too. Just looking, you kind of expect that because it's such a disparity in distance, in timing of high and low tides.
Well,
From
that's
say,
something we
Ship
just don't
Channel.
look at. We're not out there all the time just
Yeah.
monitoring that. We're just here on a
But
daily
you-
basis and
in-
sampling.
In your opinion, that there is a shorter residence time?
I would think so, yeah.
And I don't think there's a whole lot of exchange between Matador Bay and Malacca Bay.
I'm not sure about that one.
It's definitely limited.
We were here 1 October. This is a strange situation though, and, Edna had 23, 24 inches of rain in a couple of days. That whole causeway looked like a whitewater dam. I'm serious. And that bay flowed from upper Lavaca all the way to Port O'Connor. There were nothing but fire ants, piles of debris and everything, the whole bay system, even coming from the Colorado end. That wasn't a normal situation.
Some of that data also suggested, Paul, that the bays are gonna be more turbid.
The turbidity thing is wind. At the end of the day, particularly in the summer, when you get a month worth of 15, 20 miles an hour winds every day, it's just gonna stir up. And in fact, when you look at the sediments around here, I'll never forget when I first moved to Texas. It's the thing that shocked me the most was I'd never worked anywhere else. I've worked just almost everywhere in the world. The whole top, 3-4 inches of mud is alternately completely in the water column and then it falls down. It's just the wind. It's windy, it's shallow, you all know this, you've been out on boats in the bay. Particularly on windy days, it looks like chocolate milk out there. That's just the way it's gonna be, that's a wind thing. Now, you get turbidity, of course, during floods, even if there's no wind at all, but those have a tendency to be more stream-like. You can literally look like a river running down the middle of the bay, and you can see it coming out of every one of the rivers and streams and creeks. And one of the interesting things is when you look at those turbidity maps, and I know I've shown a picture of this to you before, the middle of Matador Bay can look very clear, and it's just running up and down the Ship Channel, Kawankwa, Palacios, and then of course coming out of East Matagorda, you see these big muddy plumes, but then in the middle of the bay it just looks nice and clear. So there's not a lot of circulation that, it doesn't look to me like the circulation gets, mixed up a lot, over time, except when it's windy. And of course, windy, the bay looks completely differently. The whole bay just looks like chocolate milk. So that's just the way we live here. We live with wind. Okay,
So,
thanks a lot.
Thank you very much for coming.
Thanks, guys.